I'd like to tell you about a paper we just published in EMBO journal identifying the retinal pigment epithelium of the eye as an entry route for monocytes trafficking into the retina after injury. This study was carried out in the group of Michal Schwartz at the Weizmann Institute of Science, with Benhar as lead author. So the CNS, which includes the brain, spinal cord, and the optic nerve and retina of the eye, maintains unique interactions with the peripheral immune system. These interactions are largely upheld by barriers that reside between the CNS and the circulation. Our team has previously demonstrated that recruitment of monocytes from the circulation is needed for coping with acute injuries in the CNS. In addition, we recently established that the barrier within the brain territory, which is made up by the brain choroid plexus epithelium and fenestrated capillaries, mediates recruitment of such monocytes to the brain and spinal cord parenchyma. The barriers between the retina and the circulation are comparable to those of the brain. Specifically, the outer blood retinal barrier at the back of the retina is made up of fenestrated capillaries and the retinal pigment epithelium, or RPE. Similar to the choroid plexus epithelium, the function of this barrier is maintained by tight junctions between the epithelial cells. In the present study, we explored whether upon injury to the optic nerve, which results in the death of retinal neurons, the retinal pigment epithelium could serve as a gateway for entry to the retina of monocytes that can potentially rescue these neurons. Using a model of a crush injury to the mouse optic nerve, we found that the expression of genes that encode for leukocyte trafficking determinants, including VCAM1 and others, was upregulated in the retinal pigment epithelium. Zooming in on the association of peripheral immune cells with the retinal pigment epithelium, we found that leukocytes recruited to the injured eye formed close contact with the epithelial cells. Live imaging of the injured eye showed monocyte accumulation first at the retinal pigment epithelium and subsequently in the retina, pointing to a migratory pathway of these cells from the circulation to the retina. Indeed, blocking VCAM1 after optic nerve crush interfered with monocyte infiltration into the retina and resulted in a local pro-inflammatory cytokine bias, which is generally deleterious for the survival of retinal neurons. Collectively, these results suggest that the eye, as an immune-privileged site, utilizes a mechanism of crosstalk with the immune system similar to that of the brain, whereby epithelial barriers serve as controlled gateways for leukocyte entry upon need. Our findings point to the retinal pigment epithelium as a potential site for therapeutic intervention under degenerative conditions of the visual system.